To everybody?
Yes, Dr. Vinod, that's fine. Please go ahead.
Thank you. Thank you so much. So I'm not sure how many of you are from the neuroscience or the biology background, but as I discussed during my last talk as well, I will try to keep my language in a most simplified manner so that everybody can understand what I want to convey to all of you. And you can please keep your questions for the end of the presentation. So today I'm going to talk about one of the most widely used techniques in today's world of neuroscience, which is histotoxic surgery, and one of the recent advancement in histotoxic, which is optogenetics, and how people all over the world, different scientists who are working in the field of neuroscience or developing different drugs and molecules for the treatment of different neurodegenerative disorders. So how they are utilizing this technique. So I'm going to talk about the basics, and then I will talk about some of the advancements in this field which are currently going on. So my heartfelt gratitude to Mahamana Pandit Madan Mohan Malviya for establishing this beautiful temple of learning where we are sitting today. Okay. So what you see in this slide is a white albino rat having some kind of electrode on his forehead with a light which is lightning, right, and it is connected through a wire. So this is one kind of like optogenetics approach by which we're trying to alter the different neurocircuitries in the brain of the animals, and I'll talk about that how. And this is done by using histotoxic surgery. So firstly, what is histotoxic or what is histotoxic surgery? That is, very first question to anybody who is new to this field. So it's a minimally invasive form of surgical intervention which makes use of a three-dimensional coordinate system, XYZ. We live in a 3D system, 3D coordinate system, XYZ, to locate small targets inside the body and to perform on them some actions such as ablation. We'll talk about that later. But let's talk about first this thing. Like whenever we talk about the histotoxic surgery in neuroscience, most of the people, students or even scientists, they think it's mostly about the surgery in the brain, like locating different brain regions, identifying them, locating them, and then injecting some drugs and viruses in it. But it is one second, give me 1 second. Akhilesh, इधर आइए. थर्मोसे इंस्ट्रूमेंट आ रहा है, एक बार कोऑर्डिनेट कर लीजिए उनसे. आइकन्स वगैरह थर्मोसे को इंस्ट्रूमेंट आ रहा है, प्लीज टेक इट. I'm sorry about it. So it is not only about locating or identifying the brain regions or performing the brain surgery. It is locating any region inside the body, whether it is brain, whether it is stomach or liver, kidney, or any other part of the body. And after locating that particular region in the body to perform different activities, including ablation. You want to do some kind of like ablating certain like tissue part, or ablating certain neurons or cell types, or you want to do the biopsy, for example, in case of patients who are suffering from some kind of tumor, malignant or benign. So when surgeons work on them, they use this histotoxic technique, and then they identify that tissue, whether it is present in the brain or any other part of the body, and then they take a part of piece from there by using the histotoxic surgery, and then send it for biopsy so that they can identify whether it is malignant or benign. And then people also use this technique for creating certain lesions, to develop certain models in the preclinical research so that they can study the effect of different drugs and molecules on those animal models. Then to inject some drugs, agonist or antagonist of the drug. If some of you belong to the drug discovery, you know that there are drugs which are currently in the market, which, and how they work by activating the certain receptors, or by those are known as agonists, which activate the receptors. And there are certain drugs which block certain receptors, so they are known as antagonists or inhibitors, you can say. For example, the drugs which are currently available, for the, treatment of pain, fever, like they are available as OTC, like over-the-counter medications, like aspirin is there, paracetamol is there. So these drugs are, very well-known, inhibitors of COX-1 and COX-2 enzyme. These are the two enzymes which are responsible for the generation of several pro-inflammatory, mediators like prostaglandins, leukotrienes. They are responsible for the redness, swelling, and fever. So if you have to, prevent the pain and fever, then this COX inhibitor like aspirin, paracetamol, ibuprofen, they suppress this COX-1 and COX-2 enzyme, and they inhibit this complete signaling. So we sometimes inject the agonist and antagonist of certain receptors or enzymes or hormones or certain other modulators by using this technique in different body tissues, including brain. Sometimes scientists also use this technique to stimulate the certain brain regions, specific brain regions or specific regions in the spinal cord as well. Sometimes to see what will happen if you activate a specific kind of brain region, whether it can treat certain disease. For example, deep brain stimulation these days people are using a lot, in different neurological disorders. If you stimulate certain regions which are deep into the brain, what will happen to those patients, whether they can be, treated or not. And then implantation. So, for example, in the preclinical research, in the drug discovery, we implant sometimes electrode as well. And other than electrode, we sometimes implant the cannulas in the specific regions of the brain. Cannulas are small, guide-like structures. I will talk about that in detail in the coming part of my presentation, where, we implant those cannulas in the different brain regions so that we can deliver certain drug molecules, which are agonist or antagonist or inhibitors of certain receptors or enzyme. Sometimes we also inject certain viruses, which can, which can overexpress there and lead to the, activation of certain specific neurons in the brain. Or sometimes we also inject the siRNA, which are silencing RNAs, which are used to, silence the specific neuronal or cell types in the different, brain regions or other regions of the body. The radiosurgery is another important application of histotoxic surgery. So if some patients are suffering from brain tumor, so it's very difficult, to treat this kind of patient, but we can only, prolong their life by using certain, techniques, including chemotherapy or radiosurgery, but they do have their own side effects and toxicities because non-specific, action, you can say. So in case of radiosurgery also, that is the issue, we cannot radiate the whole brain because brain consists of different kind of, cell types, including neuronal, non-neuronal, glia cell types, and maybe 10% of the brain is suffering from tumor, but 90% of the brain is good. If you radiate the whole brain, the patient may become, abnormal completely, right? So here histotoxic plays a very, very important role where, doctors, surgeons, they identify the, the exactly where tumor is located inside the brain, and then they radiate that specific part of the brain without affecting the other brain region. So this technique is very, very useful in both preclinical means in the animal models where we are, using it as a platform for the drug discovery, and also into the clinical field where doctors are using, surgeons are using this technique for the treatment of different kind of patients. So it's very useful technique. So that's what I would like to say here. Now, if you go back into the history of this, technique, where this technique, first, identified and, how it came into existence, and then it has been, evolved in previous, years. So first time in the year 1887, these are the two scientists, Victor Horsley and R.H. Clark. These are the two scientists, who were, who has designated this device first time, and it allowed for the stable placement and maintenance of the animal's head within the 3D system, which is X, Y, and Z coordinates, modeled into horizontal, sagittal, and frontal plane. Okay, we will talk about what is horizontal, what is sagittal, and what is frontal in the coming slides. And then using this device, they were able to, cut the whole brain through the cranial bone. You know that this is, brain, and then it is covered with a protective layer of the cranium, which is made up of bone, right? very strong structure, because this is very sensitive. So nature has given it a protective layer in form of cranial bone. So they have used a device, microtome. Microtome, some of you may already be aware with this device. It's a a kind of cutter, you can say, right, which can be used to cut the different sections of the brain, very thin sections, like ranging from 10 micrometer to 20 micrometer, and sometimes more than that. So they have cut the brain, animal brains basically, right, using histotaxic, and then they were able to elaborate models showing the positions of specific brain regions related to each other, as well as to the surface of the skull. Now, आप लोग ये पूछ सकते हैं कि भाई ब्रेन को काटने का क्या फायदा है, क्यों ब्रेन को काटा इन साइंटिस्ट ने? आपका जो ब्रेन है बेसिकली, या एनिमल की ब्रेन की मैं बात करूं, इसमें थाउजेंड्स ऑफ ब्रेन रीजन्स हैं बेसिकली, अलग अलग ब्रेन रीजन है, इंक्लूडिंग कॉर्टेक्स है, हिपोकैम्पस है, एंड हर ब्रेन रीजन का काम अलग है. कुछ ब्रेन रीजन ऐसे हैं जो लोकोमोशन में, मोटर कोऑर्डिनेशन में हेल्प करते हैं. कुछ ब्रेन रीजन ऐसे हैं जो आपको लर्निंग मेमोरी में, आपकी याददाश्त में हेल्प करते हैं. अल्जाइमर में वही होता है, वो ब्रेन रीजन आपके अफेक्ट हो जाते हैं, which are like cortex is there, hippocampus is there. Amygdala is another brain region which is associated with fear associated learning and memory. Okay, hippocampus is another important brain region, CA1, CA2 region, which are involved in the learning and memory. So until unless they don't cut the brain sections, very fine brain section, they may not get an idea how the brain looks from inside until they don't see it under the microscope, good quality microscope. They take the pictures, and then only they can identify, okay, these are the different brain regions and what are their relative positions from each other, so that they can understand, which brain region is present in which part of the brain, how deep one has to go, how much left, how much right to probe that brain region if somebody want to approach that brain region. So it has been possible because of this technique. Now the question, you might be having, again, some of you might be belonging to the biology background, but some of you may not be. So the question may be very genuine, why we are targeting brain? What is the need to target it? So the answer is, it's versatile approach to generate the lesions, as I told you in the, last slide. Sometime in the preclinical research, we have to develop the animal models. For example, we know that बहुत सारे पेशेंट्स हैं जिनको अल्जाइमर्स हो जाता है, याददाश्त उनकी चली जाती है पर्टिकुलर एज के बाद, कई बार यंग एज में भी जाती है, आफ्टर 40-45 years. That is a different kind of dementia or cognitive decline, but mostly it occurs in the elder age, after 60-70 years.
नहीं, वो पहले तो जाने कि क्या क्या हुआ सिचुएशन है.
Can you please mute yourself?
सही बात, 2-3 काम ऐसे है ना, उस पर वाकई लगना पड़ेगा, और हम लोगों को बस की बात बहुत मेहनत करना पड़ेगा.
आप प्लीज
नंबर कम रहेगा, फिर ग्रेड डाउन हो जाएगा, फायदा क्या है?
हेलो,
समझ लेना, स्टाफ भी करना, और भाई डेटा लिखना देना, अपना काम करना, लेटर डिपार्टमेंट सजा लेगा.
धीरेश पांडे सर, कैन यू हियर मी? प्लीज म्यूट.
सर्टिफिकेट होना ना चाहिए.
हेलो,
ये लोग कहीं से मैनेज करेंगे.
Okay, sorry about the interruption. Okay, am I audible? Hello.
Yes, please go ahead.
Okay.
Please go ahead.
Okay. So yeah, so as I told you in the beginning, that this technique is very, very useful to generate the lesions, to develop the animal models, where we can, damage particular regions of the brain which are involved in learning and memory, which are involved in motor function. जैसे कि पार्किंसन डिसीज में बेसिकली किसी पेशेंट के जो मोटर फंक्शंस होते हैं वो इम्पेयर हो जाते हैं. द पेशेंट कैन नॉट स्टैंड, कैन नॉट वॉक, कैन नॉट होल्ड अ कप ऑफ टी और कॉफी. So his body is like trembling always, hands are shaking, something like that. So to develop the new molecules or the drugs for these kind of patients, for the treatment of these kind of patients, we need the good animal models. So to do that, we use this technique, histotaxic surgery, where we generate the lesions in a specific region of the brain. For example, substantia nigra, that is the brain region which is involved in the motor function coordination. And then we deliver the experimental agents to the brain or manipulate the gene expression. And then we can also use the surgery to accurately target the deep structures within the brain through the use of a histotaxic atlas. Now, what is the histotaxic atlas basically? So histotaxic atlas is a book which has been developed, by these scientists, Watson and Taxinas. These are the two scientists, who has later on studied the brain in detail of different species, including human beings, rat, mice, or other, animal species. And they have studied, it in detail and then identified, what are the brain regions, where they are present with reference to each other, and identify their coordinates in a 3D manner. So histotaxic atlas is basically simply a kind of map, you can say, which gives you a 3D coordinates of each area with respect to anatomical landmarks on the skull. Now we will talk about what are those anatomical landmarks. These are very, very important. Bregma and lambda, we'll talk about that. Then the second important thing which is required to perform the histotaxic surgery is the histotaxic frame. So we need an instrument or a device which can help us to precisely place the experimental tools, including the Hamilton syringe or any other syringe, which can use to basically inject the different drugs, agonist, antagonist, or viruses in the different brain regions of the animals. Because we deal with neuroscience, so we are talking about the histotaxic with reference to the neuroscience. Now this is how a brain structure looks like. If you look when you cut the brain sections of rats, mice, or humans, and when you cut the sagittal section, sagittal means from front to back, sagittal sections, okay. So when you cut the sagittal sections, you can see basically the different brain regions. When you see that section under the microscope, you can see almost more than 100 brain regions you can see in the single section. And in the brain atlas, you will know, okay, this section is like 0.02 millimeter away from the bregma, and from the intraoral distance, it is around 3.82 meter. What do you see here? These black portions are LV. LV means lateral ventricles, basically. It is present on both the sides of the brain. We know that brain is, is like a structure which is divided into two hemispheres, left and right. So left hemisphere controls the right body, functions of the right body part, right? And right hemisphere controls the function of the left body part. So exactly it is a replica of on both sides, left and right. So whatever brain regions are present on the left side, they're also present on the right side. So these are the lateral ventricles, these deep structures. But this is how it looks when you cut this section at this particular position. Okay. This is another section which is like minus 2.3 millimeter away from the bregma and 6.7 millimeter away from the intraoral. Now, what I'm showing you in this slide is a histotaxic apparatus which is required to perform this surgery in animals. So this is for the rodents, basically rat and mice, which we use in our preclinical research. So in our also we have this device. If some of you might be interested to more about this instrument, this kind of surgery, and you want to get a practical exposure, how we use it in the drug discovery, in the neuroscience research, you're more welcome to visit our lab at IIT BHU. So what do you see here? There are different components in this histotaxic apparatus. Starting from number one at the top, this is macro drive screw adjustment for the dorsoventral axis. Dorsoventral axis means if you rotate this macro screw, you can decide how much down you want to go. So you have to because this is the part where you want to clamp your Hamilton syringe, which is ultimately going to enter into the brain of the animals and will be used to inject the drug solution into the brain. So to move it up and down, you have to use this number one part, which is macro screw for the dorsoventral axis. In the similar manner, there is second macro drive screw, which is number two, that is for the mediolateral axis. So for mediolateral axis means if you want to move left or right. So depending upon which part of the brain you want to inject and where it is present, according to the brain atlas, you can do the fine adjustment using this macro drive screw. Third macro drive screw is the adjustment for the anterior posterior axis. Anterior posterior means if you want to move from front to the back. So this third one, this screw is right over here. So you can decide where you want to inject, accordingly you can move it. And these coordinates are available in the brain atlas, depending upon which part of the brain you want to inject. Whether you want to inject cortex or hippocampus or anyone else. 1 second, somebody want to join, I think. Let me see. I have to admit anyone. Yeah, okay. Okay, Okay, fine. In between, I have to admit the people as well. Few people are joining in between. Okay. Okay. Then what do you see on the fourth, as the fourth option is the ear bars. These black kind of devices, this might be new to you. So these are the ear bars which are, inserted into the ears of the animals. And for your information, these animals are under anesthesia. They are not awake. So they have been given the anesthesia so that they are sleeping. They are alive, but they are sleeping. So that you can perform this surgery without any interruption and without any pain to the animals, because you have to operate on their skull and the brain. So this surgery is painful as well. But you want to make sure that they don't feel any pain and they are under anesthesia. So these are the ear bars which are used to hold the animals on this device uniformly. Okay. And then fifth one is the incision bar. This another black device here, rotated, attached to this screw. So that is to put the teeth of the animals, on the front, so that animal head is straight when you are performing this surgery. It is on the same level when you are performing this surgery. Now, I told you about two important landmarks in, last few slides. And these two important landmarks are, one is bregma and another is lambda. So these landmarks are very, very important, if you are a neuroscientist or you want to do this kind of surgery, whether in animals or you are a surgeon, you want to operate on human beings, then these landmarks are very, very important and you should be familiar with this. If you look, this is just a picture to tell you, this is just a replica of, what do you see here. So B here represent the point which is known as bregma, and L is the another intersection which represents as lambda. So this is almost 9 millimeter away from bregma in case of adult mice. In humans, this distance may vary. Rostral part means frontal part where the nose is there, and the occipital means back side of the head basically, which is towards the ear. So this is rostral and this is occipital. So bregma is more towards the rostral part basically. So what is the bregma basically? You can easily see this is like a plus sign. So it is an intersection of two sutures. So whenever you open the, or when you put a cut on the skin of the skull skin of the animal, and then when you put a cut on the muscle which is there, and then if you expose it, you will see a intersection where two lines are meeting each other. So this is the intersection of the intersection of the sagittal suture, or SS you can call it, SS, which is running from top to bottom, and then another is the coronal suture, which is running from left to right, CS, like this. So it is intersection between the sagittal suture and the coronal suture, and the point where they are meeting each other, coronal suture and sagittal suture, is known as bregma. Now, why this point is important? This is important because this is considered as the coordinate which is origin of all other brain regions. So this means this is acting as a reference point. जैसे हम बोलते हैं कि हम यहाँ से रेस स्टार्ट करेंगे, तो यानी कि this is the origin point. So this is the origin point for all the different brain regions where all the coordinates x, y, and z are 0, 0, and 0 millimeter. Okay. Then there is another important landmark which is known as lambda. So this is the point which is located on the midline. It's a Y-like structure, inverted Y you can say. You can see it here, right? It's like sagittal suture is running, and then it is making an inverted Y. So at the base of the triangle, it is a kind of triangle basically, right? Formed by the lambdoid and sagittal suture. So this is the lambdoid suture, and then this is sagittal suture basically, this one. And this is lambdoid. So this point is known as the lambda. Now you may ask that why this lambda is important, why we should bother about it, why it is so important. It is important because when you perform the surgery, and if the this bregma and lambda, they are not at the same coordinate, then what will be happening? You are thinking that you are injecting some of the brain region which is deep down, and you are injecting, for example, hippocampus, C1 region. But actually you are not injecting C1 region, you are injecting somewhere, on the top of that or on the bottom of that basically. So your injection may not be accurate if the head of the animal is not straight. So this coordinates tell you bregma and lambda, if they are on the same level, then only that will tell you that the head of the animal is completely straight. So after this, if you perform any injection, macro injection in these animals, it will be accurately delivering to that brain region only. Now, before performing this kind of surgery, what are the requirements which one has to do actually? So first important thing is to prepare the animals, because we deal with the different models in drug discovery. So yeah, it's more about the animal like surgery. So we prepare the animal and then before that we have to sterilize all your equipments. And how you're supposed to do that? You are supposed to keep your all the equipment, like blades is there, cannulas are there, scissors are there, or any other equipment where clamps are there, right? All these things are supposed to be kept in the 70% ethanol 1 day before the surgery, and keep it there for overnight so that they are completely sterilized, and then keep them in dry air so that alcohol gets evaporated and they're sterilized. After that, you have to take the animals, rats. If you're performing the surgery on rat, you can use mice as well, which are around 25 to 30 grams. And we generally prefer the animals which are adult and healthy basically. Sometimes you have to go for small animals as well, depending upon the objective of your study. But in most of the study, we use the adult animal which are healthy, okay, which are almost like 8 to 12 weeks old. And then we go for the anesthetizing the animal. And we can anesthetize the animal by using certain anesthetizer, like these are ultra short acting drugs, isoflurane, continuous supply of the isoflurane. These are ultra short acting anesthetics, but you are continuously infusing it in this respiratory system so that they are completely under anesthesia. Or you can also use a combination of ketamine and xalazine. Ketamine is an NMD receptor antagonist, and xalazine is a skeletal muscle relaxant. So you can use it in a perfect dose combination, which is reported in the literature, to make sure that your animal is under anesthesia for at least 45 to 60 minutes while you perform this surgery. And then after anesthesia, your job is to shave the head area where you want to do the surgery. Simply, जब डॉक्टर भी सर्जरी करते हैं पेशेंट्स में, तो पहले बाल वगैरह सारे क्लीन करते हैं. In the similar manner, when you want to do the surgery animals, you have to shave the head area from the ears to just in between the eyes with an electric razor. And then after that, you are supposed to put the rats on the heating pad, warm pad basically, right? Why? Because whenever animal or any other living being is under anesthesia, their body temperature drops. And if it is dropping below a certain point, then animal may feel cold and sometimes may die as well. Okay, so it is very, very important to maintain their body temperature. So that can be done by putting the heating pad underneath their body, and you can maintain the body temperature at 37 to 38 degrees Celsius almost. And you can also use the oximeter to check whether their blood oxygen level should not drop below 90%. So these are the vital signs which you have to monitor, and which will tell you whether animal is alive or not. Because this kind of surgery, even if you have done the overdose of anesthesia, the animal may die. You don't want that, right? You are doing a survival surgery where you will do the histotoxic surgery and then will wait for the awakening of animals so that you can use them further for drug testing. And then you also need to apply the eye cream on both the corneas to avoid the dehydration. Because, and what we do frequently, when we are talking to each other or whenever we are doing our routine work, we keep on blinking our eyes. Why do we blink our eyes? It helps us to keep our like eyelids or cornea wet through the lacrimal fluid. But when the animal is under anesthesia, they cannot blink their eyes, and it will lead to the dehydration and damage in their cornea. So it is very important to apply some eye cream. Give me 1 second, I have to admit someone, I think. Okay, sorry about this interruption between. People are joining actually. We need to admit them. Okay, so yeah, and then we can continuously monitor the rat's temperature with a rectal thermometer. That is another vital sign, to make sure that they are under anesthesia and on time. Okay. Now, after you have done the animal preparation, you have to go for the surgery. And this is how this setup looks like. The apparatus I showed you in the previous slide, histotoxic apparatus, it looks like this actually. So this is the histotoxic apparatus, and this is the nose clamp, and these are the ear rods, which are placed in the left and the right ear. And it is very, very important, otherwise animal will not be stable on this platform. And when you do the surgery, when you are trying to put this incision on the head of the animals will drop actually. So you don't want that, right? You want this surgery to be done precisely and accurately. And then you also have to inject the analgesic, which is a painkiller, so that you want to make sure that they should not feel pain during the surgery and after the surgery. And then place the animal in the histotoxic frame. And we need to check the loss of paw reflex with a pinch tweezer. So हम लोग क्या करते हैं, pinch tweezer से उनके paw पे हल्का सा pinch करेंगे, just to confirm that they are under deep anesthesia. Otherwise, what will happen? You have given them the anesthetic, you have also given them the painkiller, but the animal is not under anesthesia, and then you're putting an incision. You can just imagine how painful it could be for the animals. So considering all ethical guidelines in your mind, then only you should do this kind of surgery. You have to make sure that the reflexes, are not there, and after that only you should go ahead for the surgery. And then place the head in the nose clamp. This is the nose clamp here, right over here, this one. And then insert the ear bars carefully. And then you can secure the Hamilton syringe. So Hamilton syringe are very, fine syringes by which you can deliver the macroliter, amount of any drugs, agonist, antagonist, into the brain of the animals. And then you can clean this, shave area with disinfectant like 70% ethanol, and also apply the povidone iodine, which is a tincture iodine basically, to make sure that animals should not get infection after the surgery during the recovery phase. And then after that you make an interior posterior incision. So anterior posterior means from the from the top to the bottom basically. So here you can see this is a surgical blade, and the person is making a anterior posterior incision. And then make an incision of about 2.5 centimeter on the midline of the skull, going from between the eyes until the back of the ears. And then you can use the bulldog clamps. These are known as bulldog clamps, so that you can, spread the skin and the muscle and see the bregma. Your first job is to locate the bregma and the lamina on the skull of the animals. This is present in all the living beings, not only rat and mice. It is also present in the humans as well. So they act as a landmark which can be used to identify the other brain regions. And if there are any conjunctive tissues, they can be removed, and with the cotton swabs, if there's certain bleeding, you can clean it with the help of cotton swabs and clean the area to expose the skull surface. And then after that, you can swing the stereo arm. Stereo arm is the arm in which Hamilton syringe is attached above the skull, and carefully lower the tip of the syringe needle to measure the Z position. Z position means the dorsoventral, means how deep you want to go into the brain. And then you have to measure whether lambda and bregma, they are both at the same level or not. Then only you can judge that, okay, the head of the animal is straight. You cannot judge it with the naked eye. With the naked eye, you may be thinking that, okay, the animal head is straight and injection will be perfect, but this is not the case. You have to accurately measure in millimeters, even in points. If it is 3.4 mm, you can do plus minus 0.01, but not more than that. So you have to make sure that, okay, lambda and bregma, they are at the same level when you're measuring it with the dorsoventral position. And if there are certain, differences, you have to adjust the position of the nose clamp to make sure that the lambda and bregma are at the same Z position, same Z plane. And that way, your injection will be in the right position. And then after that, you have to measure the coordinates for the injection site. Now, you want to inject one of the brain regions in 1,000 brain regions which are present deep down there. You can just imagine, how difficult it is, and how precisely it can be done by using this technology. There are thousands of brain regions, but you just want to inject in one brain region, which is so small. That's why you cannot inject a big amount of, big volume of the drug. You can hardly inject, 2 to 3 microliter or 5 microliter, but not more than 10 microliter, because brain regions are very tiny, very small. If you try to inject more than 10 microliter, these brain regions may swell and burst because of the excessive volume of the fluid. So whatever drug, agonist, antagonist you want to deliver in the brain regions, you have to dissolve in this amount of drug volume only, which is hardly 1 or 2 drops, not more than that. And then these are the coordinates X, Y, and Z, which are designated as anterior posterior. So what you can see here, basically, this is the anterior posterior, like, from the bregma, how much you have to go, like towards the eyes or towards the back. So if you go towards the eye, this is plus anterior posterior. If you go towards the ears, it is negative anterior posterior. Okay. Then second coordinate is a medial lateral. Medial lateral means you can go either to the left or to the right. If you go to the left, it will be plus. If you go to the right, it is minus. And then last one is the DV. DV means dorsoventral, means after you identify this, brain region on the skull, you have to drill it so that you can go deep into the brain. So that is the dorsoventral. So after drilling some part of the bone where you want to inject, where your brain region is present, you have to put the Hamilton syringe and then you have to go deep down. And then once you identify this anterior posterior medial lateral, for example, it is mentioned like 3.2 mm. So you will go 3.2 mm from bregma, like here. And then if it is medial lateral 0.2 mm, you will go like this. So now this is the point, for example, where your brain region is present. So this point you will mark with the marker, permanent tissue marker. And then you use the driller and, drill at this position. Carefully, slowly. You have to be very, careful while drilling, because if you don't drill this bony part carefully, you may damage the brain of the animals, and this is not what you want. Okay. And then once you drill the hole, you can just have to be careful, otherwise there will be brain damage and bleeding. Then bring the stereo arm over the hole, this drill what you have done, and position the syringe, Hamilton syringe, on the top of the hole, and then carefully lower the syringe at very low speed, because as I mentioned, these brain regions, you are penetrating the brain, which is one of the most sensitive region of the body, right? So you have to approach the brain region because you are going inside the brain by using this Hamilton syringe. So you have to do it very slowly at a speed of around 0.2 millimeter every 20 seconds, which is very, very slow, because you don't want to damage the brain regions. There will be certain damage, but it will not be that much until it reaches the required depth, which will be measured by the dorsoventral axis. Okay, fine. Okay, so after the syringe is in correct position, you have to press the plunger. Plunger means the basically injection. जैसे हम लोग आपने देखा होगा hospital में. I don't know how many of you are from the biology background, but those who are not, right? If you want to inject something, you have to push the plunger from the top. Then only you can inject the drug, or anything, into the body of the patient, right? Similarly here, you want to inject the drug solution into the brain, which is a very small amount, right? So you have to do it very carefully, very slowly, and the rate should not be more than like 0.4 microliter every 30 seconds. So you can just imagine, if it is 4 microliter, that means it will take 30 into 10, that is 300 seconds, which is almost 5 minutes. So 5 microliter, which is just a single drop, you have to take at least 5 minutes to inject into the brain of the animals. And then you wait for 2 minutes. After you are done with the injection, you have to hold on for a few minutes. Why you have to wait? Because sometimes when you are injecting something into the brain and if you're immediately withdrawing, what will happen? There could be a backflow. You don't want that backflow. Okay. And then slowly retract the stereo arm at a very slow speed, because slowly you have to reach the brain, penetrate the brain, and then slowly you have to withdraw from the brain as well. Okay. And check that there is no backflow of the fluid after removing the syringe. You have to clean the incision with 70% ethanol, and have to apply the povidone iodine antibiotic, and apply a small amount of bone wax to seal the hole so that there is no backflow later on. And then you can suture the borders of the wound, bringing the, so you have put the incision on the muscles. So now you have to stitch it back. जैसे कि हम लोग कपड़े से लेते हैं करके सुई धागे से. In the similar manner, surgeons also do the same thing in patients. When they do operate the patient, they cut the muscles, skin, and then they suture it back. In the similar manner, you have to bring both the lips of the wound together and then tie it by using the surgical suture and making a triple knot so that it doesn't open later on. and then apply the antibiotic ointment so that you can prevent the infection in these animals, and then return the animals to the cage and place it on top of a small heating pad. This is very, very important, and then you have to wait, until the animal recovers from the anesthesia. Sometimes, most of the time, the students, who are doing PhD or postdoc or masters, when they are done with the surgery, they just put the animals in their cage. Jahan pe rehte hain animals basically, right? And they don't care, whether their nose is, where. Sometimes the nose goes beneath the, bedding material, and they're not able to breathe. Sometimes the animal is dying not because of the surgery, but because of the anesthesia. They are under anesthesia and their nose is deep into the bedding material. So you have to be very careful while you are putting the animals, and then watch them carefully whether they are breathing or not. And then it is very, very important, that once you are done with the surgery, you have to provide them a very good post-operative care, at least for a couple of days till they are recovered from the signs of surgery. So daily you have to check your animals, especially in the first 3 days after surgery, and then notify immediately if there are any problems with these animals, such as lack of movement. अगर आपने गलती से brain damage कर दिया है बहुत ज्यादा, उस केस में उनका motor function impair हो जाएगा animals का. They won't be able to walk in their home cage properly. If you see any inflammation in the wound, like swelling है, redness है, या कोई infection है, that also has to be properly taken care. If you see the signs of lordosis, now what is lordosis? Lordosis means arch back. इनकी जो back है जब वो चलेंगे तो it is like a hump basically, जैसा camel hump होता है. So lordosis is a sign of pain basically in these animals. That means these animals are suffering from pain. So it has to be taken care properly. The animals should be given analgesic, the painkiller, so that their pain can be suppressed. Now, if you see there are certain stitches जो आपने लगाए थे surgery के time पे, after surgery, if they are open, then again you have to put them back under anesthesia. Otherwise, what will happen? The infection will spread in the, those muscles and tissues, and you don't want that with your animals. Now, animals that show overt signs of sickness, कई बार बीमार अगर होते हैं animal, infection of a wound, loss of body weight, that is one of the most important parameter to understand whether your animals are sick or not. If they're healthy adult, generally their body weight should be maintained if it is not increasing, because they are already adult. So it will not increase drastically. It should be maintained over a period of days or weeks. But if it is reducing drastically, like one or the other day, that means your animals are severe sick or other signs of reduced well-being. Then you need to provide the special or extra care to your animals by providing them extra dose of analgesic, which is painkiller, or a mixture of powder food and water. कई बार एनिमल के पेन होता है, जैसे हम लोग भी बीमार होते हैं कई बार, ये सर्जरी के बाद डॉक्टर क्या कहता है, इनको खिचड़ी खिलाओ या दलिया खिलाओ, क्योंकि सॉलिड फूड खा नहीं सकते, चबा नहीं सकते, दर्द होगा. Same things with animals as well. After the surgery, you cannot expect them to chew that hard pellet diet. They cannot chew it. It's very hard because they will use their teeth and it will create pain in their skull or brain. So in that case, you can do one thing. You can powder the pellets and then mix it with water and then put it inside the cage, so that they don't have to struggle to get their food. And that way your animals will be recovered very quickly. And the rat doesn't show any improvement after these interventions. कई बार ऐसा होता है कि animal बहुत ज़्यादा बीमार है, बहुत ज़्यादा infected है, so you won't see any improvement. Or loss of body weight is more than 15 to 20%. That means if the body weight of the animal was 300 grams and animal has lost almost 60 grams of the body weight, which is 20% of its original body weight. In that case, that means animals are severely sick. These kind of animals are supposed to be sacrificed with an overdose of the anesthetic form. And this is known as a human endpoint, because they are not recovering, they are suffering because of some surgical procedures or infections or some other issues. So these kind of animals should be humanly sacrificed under deep anesthesia. So we also call it euthanasia. And then rats usually need to recover for at least 7 days, 7 to 10 days before you start any behavioral studies on these animals. If you're working on learning memory, like Alzheimer's disease, then there are certain behavior experiments by which you can check whether the learning and memory of animal is good or bad. In the similar manner, if you're working on Parkinson's disease, which is another neurological disorder, जहाँ पे पेशेंट्स के हाथ पैर कांपते हैं, मोटर कोऑर्डिनेशन इंपेयर हो जाता है, उसके लिए अदर बिहेवियर टेस्ट हैं. तो वो सब करने के लिए आपको लेकिन वेट करना पड़ेगा जब तक आपका एनिमल थोड़ा सा रिकवर ना हो जाए 7 to 10 days. If we talk about the application of this histotoxic surgery, it has a, like I would say a huge application. They act as a, like biomimicry of the clinical model. Why I'm saying biomimicry? Just, I give you an example, if you want to mimic the model of Alzheimer's disease in animals, in rat, mice, or even in higher species like monkeys, then also, you can use this surgery. एक बायोमार्कर है अल्जाइमर डिसीज की अगर हम बात करें एमलाइट बीटा. आई डोंट नो सम ऑफ यू माइट बी फ्रॉम द बायोलॉजी बैकग्राउंड एज वेल. सो एमलाइट बीटा बेसिकली एक प्रोटीन है जो कि अल्जाइमर के टाइम पे ब्रेन के सर्टेन ब्रेन रीजन्स में क्या होता है उसका प्रोडक्शन बढ़ जाता है, स्पेशली एमलाइट बीटा 42 का. उसकी डिफरेंट सब फॉर्म्स हैं, बट एमलाइट बीटा 42 जो है वो काफी टॉक्सिक है. Why it is toxic? Because it is insoluble in nature. So brain के जो न्यूरॉन्स हैं उसको एक्सक्रीट आउट नहीं कर पाते, टेक आउट नहीं कर पाते. तो what will happen that amyloid beta will keep on accumulating inside the brain. और उसके स्टिकी नेचर की वजह से क्या होगा, आपके जो बाकी जो ग्लियल सेल्स and neurons they will start having the issues because of the formation of neurofibrillary tangles, A beta plaques basically, right? So that will lead to the learning and memory impairment. So by injecting this amyloid beta peptide, we can create this model in the animals as well, in rat and mice. So this is known as the biomimicry of the clinical Alzheimer model, which is evident in the human patient, जो humans में होता है. और हम ऐसा क्यों कर रहे हैं? ताकि जो भी ड्रग्स जो हम ह्यूमंस में डायरेक्टली चेक नहीं कर सकते, सिंपल सी बात है, जो भी ड्रग्स आज मार्केट में है ये बिकॉज ऑफ दिस एनिमल मॉडल्स. You cannot test any new molecule directly on the humans. Why? Because you don't know whether they are safe or not. So I should tell you this, one case study, of one drug, which is known as thalidomide. Some of you might be having an idea. So this drug was, was introduced into the market, like few hundred years back, and it was in Europe especially, and not much studies were done on it, like especially safety studies. So this drug was especially, being marketed for the pregnant women to control their morning sickness. जो उनको मॉर्निंग में जो वॉमिटिंग और नॉजिया वो सब होता है उस सबको सप्रेस करने के लिए ड्रग मार्केट में आई थी कई साल पहले, थैलिडोमाइड. बट व्हाट हैपेंड, उसके ऊपर बहुत ज्यादा स्टडी नहीं की गई बिकॉज रेगुलेटरी गाइडलाइंस उसमें काफी रिलैक्स थी. रेगुलेटरी जो मार्केट है, जो एफडीए गाइडलाइन है या डीसीजी जो यूरोपियन मार्केट की गाइडलाइंस हैं, दे वर नॉट वेरी स्ट्रिंजेंट. And drug came to the market without much studies basically, on the animal models. Okay. So what happened actually, after a few years, it was found that thousands of children, the newborns throughout the Europe, throughout the world, were born with a disease which is known as phocomelia. Now you might be thinking, what is phocomelia? Some of you might be from the biology or the medical background. So phocomelia basically is a disease where a child is born without limbs. उस बच्चे के हाथ नहीं है, पैर नहीं है. अगर पैर हैं तो seal like हैं, जैसे मछली की तरह ये होते हैं. And there are a lot of physical abnormalities and also mental abnormalities in these children because of that drug thalidomide, because that drug was teratogenic in nature. Teratogenic means the drugs which affect the fetal development inside the mother body. So अगर वो ड्रग मार्केट में अगर प्रॉपरली सेफ्टी स्टैंडर्ड्स के बाद आती तो शायद ये सारे इश्यूज नहीं फेस करने पड़ते. आज भी वो बच्चे सफर कर रहे हैं, दे आर स्टिल देयर इन डिफरेंट पार्ट्स ऑफ द यूरोप. फॉर्चुनेटली अमेरिका में एक साइंटिस्ट थी एफडीए में, उनका नाम अभी मैं रिकॉल नहीं कर पा रहा हूँ. She said this drug cannot be introduced into the American market, in the US market, because we don't have enough data on its toxicity and safety basically. And American, government did not allow, that drug to enter into the American market, into the clinic basically for people use. So they were fortunate to save from that tragedy, and that FDA officer later on got the President Medal, from the US government later on to save the American population from that tragedy. So that was one of the well-known tragedy where thousands of kids have suffered that time. And they are still suffering throughout their life. You can just imagine the situation. So that is the thing, why this, drug discovery, animal studies plays a very, very important role. So before you bring any drug into the market, you have to test them in the animal model. So that's why we are doing the biomimicry of the clinical models. Now, second is understanding the complex biological neurocircuitries. Now, in the human system or in the human beings, we cannot subject them directly into the experiments, right? We know that, okay, animals or humans are different from each other. They're not exactly same, but at least some of the physiological or anatomical features are, similar at least to some extent. So to understand the complex, like the hormones are same, neurotransmitters are same, they might have some differences in percentage, but most of the things are same, right? So we try to understand, what are those biological neurocircuitries which are altered during the disease condition, and if you give certain drugs or molecules, can be treated. Identification of the physiological pathways, like which receptors are enzyme, which enzymes are involved, what hormones are being altered, and then finally, it plays a very, very important role in the discovery of new drug molecules, which are effective as well, but before effectiveness, they should be safe to the patient. They should not be toxic. That is the first priority. That's why whenever we talk about the clinical trials means testing of drug in the humans. That is always done when extensive studies are conducted already in the animal models, in vitro system and the in vivo system. So then only this drugs can be tested in the clinical trials, that in testing on the humans. So the first, I already told you, the first application is to induce the lesions, right? So this we have already discussed. We can inject certain toxins, we can do a transaction, we can do the photoirradiation in the animals, or we can temporarily inactivate a particular brain region, through macro injection of neuromodulator, or we can inject some receptor antagonist. So receptor, I'm not sure whether you have an idea or not, but it's just like a lock and key mechanism, right? Whenever any drug is being given to us, जैसे कि मैंने आपको एग्जांपल दिया था एस्पिरिन का, पैरासिटामोल का, वो कैसे काम कर रही है? वो बेसिकली कॉक्स1 और कॉक्स2 जो एंजाइम होते हैं उनको इनहिबिट करती है. ऐसी बहुत सारी ड्रग्स जो हम खाते हैं डायबिटीज ठीक करने के लिए या कैंसर के लिए या वो बहुत सारी डिसीज में हम लोग जो दवाइयां खाते हैं डे टू डे लाइफ में भी, वो कैसे काम करती हैं? वो रिसेप्टर्स को एक्टिवेट करती हैं. उनके रिसेप्टर्स कहाँ प्रेजेंट है? हमारी बॉडी के डिफरेंट टिश्यूज में प्रेजेंट है. So sometimes they activate these receptors, sometimes they inhibit these receptors. So we do those kind of things also by using this technique. So this is one of the I would say publication, one of the very early publication from my PhD research work when I was a student, in 2008 around, almost like I think 14-15 years back, So where we utilize this technique, histotoxic surgery for studying the effect of tocotrienol. Tocotrienol is one of the isoform of the vitamin E actually. So we studied the effect of this molecule in the animal model of intracerboventricular. So ventriculars, these are the brain regions which are present, in humans and animals as well. And where we injected this streptozotocin, which is one chemical which you inject by IP route, then you can induce a diabetes in the animal. But if you inject directly into the brain, it will lead to cognitive dysfunction. And then we published the paper that time in 2009 in Pharmacology, Biochemistry, Behavior, one journal. The second important application of this technique is the tracing of the novel neural pathways. Now we understand that everything is connected at a neuronal or the non-neuronal level, from the brain up to the spinal cord and into different tissues. Now to understand, for example, in this case, there are different kind of neurons which are present. First order neurons, for example, in case of taste buds, we can taste different kind of food. How? With the help of taste bud. Why we are able to do that? Because we have the neurons which are present on the taste buds. So these are the primary order neuron or the first order neuron. Now how they are connected? They are connected with the sensory ganglia, which are known as the secondary order neuron or second order neurons. And now these neurons are connected to the third order neuron or the higher neurons which are present in the brain stem. So you can see how the system works. आप जब कोई चीज छूते हैं या आपको कहीं पे गर्म ठंडा महसूस होता है तो वो भी ऐसे ही होता है, because primary neurons are present in the fingertips basically, right, or the foots. Now secondary neurons are present in the dorsal root ganglia or in the spinal cord, and tertiary or the third degree neurons are present in the brain basically. So all these neurons, they are connected to each other. You can just see where is the brain and where is the hand, but they both are connected to each other. How? Through these pathways, different neurons. First order, second order, and third order. Now to find out the connectivity between these neurons, we can use this histotoxic technique, where we can inject specific dyes in one region of the brain, and then we can trace it, if it is leaking to particular, if it is present in particular neuron which are present in the DRG, spinal cord, or certain other tissues or organ basically. This is how we try to identify their relationship to each other. Now, collection of biological samples, that is another very important application of histotoxic surgery. Now, generally we don't do it in the human beings in the clinic, but sometimes, when doctors or surgeons are operating on the brains of the patients, or when they are doing some kind of, operation in the spinal cord, that time they, collect the cerebrospinal fluid. Why it is important? Because they wanted to understand what is present in the cerebrospinal fluid. ये भी एक kind of biopsy है. टिश्यू में हम बायोप्सी में क्या करते हैं, हम टिश्यू ले लेते हैं. यहाँ पे क्या करते हैं, CSF को निकाल के उसमें स्टडी करते हैं what is present in these biological samples. The same thing preclinical scientists do in the animal models. So for this they use a macrodialysis probe. Now this macrodialysis probe, is very expensive because it has different, mechanism to, filter the macromolecules, from the cerebrospinal fluid, because there are different proteins and other things which are present. But if it doesn't filter everything, then everything will come out in the CSF. That is something which you don't want, right? And then to implant this macrodialysis probe into the animals, you need to perform the histotoxic surgery. So what do you see here? This shaft can be, inserted into the brain of the animals at a desired location, from where you want to collect the cerebrospinal fluid. What do you see here is a cage where animals are placed, and then there is a syringe basically. So one is the inlet. From inlet you can inject the artificial cerebrospinal fluid, and from the outlet you can collect the cerebrospinal fluid from the brain, and then immediately it will be collected in a collector which is maintained at a minus temperature, so that whatever neurotransmitters you want to measure, whatever other modulators you want to study, they should not degrade, because they are very transient, these neurotransmitters. If you don't fridge it immediately, they will be degraded very quickly. Okay. Then what you can do the sampling by using this, a macrodialysis technique using histotoxic, and then you can do the real-time measurement of changes in neurotransmitter. So you can connect that collector with the HPLC or the LC-MS. Now, what is the HPLC and LC-MS? These are the tools or the techniques by which you can measure the levels of neurotransmitter in the real time. कितना डोपामिन प्रेजेंट है, कितना एसिटाइलकोलिन प्रेजेंट है, कितना सेरोटोनिन प्रेजेंट है. हो सकता है कि आप लोगों ने सुना है न्यूरोट्रांसमीटर के नाम, बट जिन लोगों को नहीं पता है, these are the neurotransmitters which are responsible for most of the things in our life. For example, serotonin की अगर बात करें, serotonin is a neurotransmitter which gets dropped during the depression. जो पेशेंट डिप्रेशन के मरीज होते हैं उनमें सेरोटोनिन की कमी हो जाती है ब्रेन में, इसलिए उनको वैसी ड्रग्स दी जाती हैं जो सेरोटोनिन के रीअपटेक को इंक्रीज करते हैं, लाइक सेलेक्टिव सेरोटोनिन रीअपटेक इनहिबिटर. Dopamine is a neurotransmitter which is involved in the reward circuitry. कोई प्रमोशन मिलता है, कोई प्राइज मिलता है, या फूड या any other motivational factor, this dopamine gets increased. But कई बार इसकी वजह से एडिक्शन भी हो सकता है if it goes to the other circuitry. Acetylcholine is another neurotransmitter which is involved in the learning and memory. So these neurotransmitters, they play a very, very important role in different, biological functions in human beings and in animals as well. So we can measure these neurotransmitters, how they are modulated during the disease condition and after the drug treatment in animals and in humans as well. We can also measure the time dependent changes, like ड्रग देने के आधे घंटे के बाद क्या इफेक्ट है, 1 घंटे के बाद क्या इफेक्ट है, 2 घंटे 4 घंटे के बाद क्या इफेक्ट है. ये सब कुछ हम लोग रियल टाइम मैनर में इसको हम डिटेक्ट कर सकते हैं, एनालाइज कर सकते हैं, क्वांटिफाई कर सकते हैं. And then apart from that, we can also go for the enzymatic activity, electrolytes उसमें कौन-कौन से प्रेजेंट है, हार्मोन्स प्रेजेंट हैं कि नहीं है, कौन से हार्मोन्स बढ़ गए या घट गए, ये सब कुछ हम कर सकते हैं by using this technique. Then again, rodent model of human disease, ये already हमने discuss कर लिया, what I told you, like by injecting amyloid beta for studying the Alzheimer disease, we can inject 6-hydroxydopamine. This is also one chemical which induce the damage in the substantial nigra region of the brain, or alpha-synuclein basically, right? So these are the neurotoxins basically which can, damage the certain regions of the brain. In this case, the regions which play a very, very important role in the motor function, and they can induce the Parkinson-like symptom. Similarly, stem cell transplantation is, one of the emerging field. People are working on it from last few years. So people are using neural stem cells because we know that stem cells are the cells which can be differentiated into any kind of cell type. They are the mother cell, you can say, right? They can form the cells of any body types like liver ki cell ho, kidney ki ho, brain ki ho, kahin ki bhi cell ho, they all are originated from the stem cells. So now people are, trying to study these stem cells, for the regenerative therapy, for the treatment of several illnesses, including neurodegenerative disorder. So people are using histotoxic surgery to deliver these neural stem cells directly into the brain region. Okay. Then another interesting application is the remote control animals. You might not have heard this technique before, right? And we also call them biorobots. Why we call them biorobots? Because we can control them remotely, okay, by humans. So here we are controlling them by implanting the microelectrode, which are implanted into their brain regions, and then we can stimulate those electrode, or the reward center. There are certain regions, like nucleus accumbens, ventral tegmental area, and few other regions which plays a very, very important role in the reward activation, reward circuitry. So they can be used, for that purpose. In 2002, a team of scientists, at the State University of New York, they remotely controlled the rats from a laptop up to a distance of almost 500 meters, which is a significant achievement. And people are still working, in this area. And then, next, interesting, I would say application of histotoxic surgery is the optogenetics, which is, widely used nowadays, in case of, preclinical research in the neurological, drug discovery. So, so what is optogenetics basically? It's a neuromodulation approach which controls the neuronal activity using the light. So this term is made up of two words: opto. Opto means light. Genetics means the genes basically. So by using the light, basically we are manipulating the genes. Any means we can activate certain genes or we can silence certain genes. Anywhere. Here we are talking about the brain. So how we do that? We use bioengineered light sensitive proteins. Now you might have heard about the channel rhodopsin or holorhodopsin, right? Right? They are present in the rod and cone cells as well. Eyes. This is some data basically. Rhodopsin. So channel rhodopsin is basically responsible for the activation of the neurons, and holorhodopsin is another protein which is responsible for silencing of the neurons, right? And now the very interesting thing is that you can use the lights of certain wavelength. So if you use the blue light, we are going to activate the channel rhodopsin. And if channel rhodopsin will be activated, what will happen? It will activate the certain neurons. In the similar manner, holorhodopsins can be activated by using the light of another wavelength, which is yellow light. So if you switch on the yellow light, what will happen? Yellow light will activate the holorhodopsin, but the function of holorhodopsin channels is because they are operated by the chloride ion channel. There will be a lot of chloride influx, lot of chloride will go inside the neurons, and that will silence the neuronal activity. Okay, so that light is used, or that wavelength of light is used for silencing the neuron. So what is the most beautiful part in this technique? You might be thinking, why I'm talking about this. You can activate or silence any neuron in any part of the brain, specifically. So कोई भी ड्रग जब हम इंजेक्ट करते हैं हिस्टोटॉक्सिक सर्जरी कैनुला इम्प्लांट करके, तो उसमें क्या हो रहा है? ड्रग तो आपके सारे ब्रेन रीजन में जा रही है और डिफ्यूज भी हो रही है। उस ब्रेन रीजन में जा रही है तो क्या कर रही है? जितने भी रिसेप्टर्स हैं, जितने भी न्यूरॉन्स हैं, वो सबको अफेक्ट कर रही है। बट हियर इफ यू वांट टू selectively activate a specific neuron type, in that case you can use this technique. Okay, and the beauty is that you can do this within a period of millisecond temporal accuracy, not even second, milliseconds. So this is how this technique works, just basics of this technique. So mainly 6 steps are involved. The first step is that we use a genetic constructor, okay, where we are using the promoter, to drive the expression, of this, light sensitive ion channel. And there's a gene which is present, which encode for the opsin gene, basically, right? It's a light sensitive ion channel opsin, which is present in the rod and cone cells as well in our eyes. And then we insert the construct into this virus by using the recombinant DNA technology. Now, why virus? Because virus has a capability to replicate. And once it will replicate, it will make the millions of copies. So that means this gene will also replicate along with the virus. And when you inject this virus into the brain of the animal by using histotoxic technique, the opsin, whether it is rhodopsin or whether it is channel rhodopsin, any opsin, to activate the neuron or to silence the neuron, it will be expressed in the targeting neuron. किसी भी न्यूरॉन में, चाहे वो डोपामिनर्जिक न्यूरॉन्स हो, सेरोटोनिन वाले न्यूरॉन्स हो, या एसिटाइलकोलिन वाले हो, या गाबर्जिक हो, ग्लूटामेटर्जिक हो, एकदम हो सकता नहीं हो आपके लिए, बट ये बहुत अलग अलग टाइप के न्यूरॉन्स बेसिकली प्रेजेंट होते हैं ब्रेन में, और आप किसी भी न्यूरॉन को एक्टिवेट कर सकते हैं और साइलेंस भी कर सकते हैं बाय inserting into a specific neuron. And then you can insert the optrode, which is fiber optic cable, but these days you can do it by using Wi-Fi as well. This fiber optic is not required in the advanced technology. And then it is connected with certain instrument where you can decide what kind of laser light to give to these animals, whether blue light or the yellow light. And once you activate this, it will open the ion channels. It can be a channel rhodopsin, which can open the sodium ion channel, or if it is hollow rhodopsin, it will open the chloride ion channels. Sorry. And then you can do whatever you want to do with these animals. After activating the ion channels, you can measure their behavior activity. Behavior instruments में आप इनको चेक कर सकते हैं, इनका चलना फिरना उठना बैठना, या कोई टास्क को याद रखना है तो ये याद रख पा रहे हैं कि नहीं रख पा रहे हैं, या फिर हम इनकी इलेक्ट्रोफिजियोलॉजिकल रिकॉर्डिंग्स भी कर सकते हैं ब्रेन से या और किसी बॉडी टिश्यू से. So all these things are possible. But there are certain challenges, for translation of this technique, optogenetics from bench to patients. We can do it in animals, but doing it in humans is still, a challenging task. Why? Because delivering opsin gene is invasive. We have to, reach to the brain. Humans के brain में it can be risky because you are injecting. कोई भी human नहीं देगी आपको infection in humans. Into deep brain regions would require optic fiber implantation, which may cause trauma to the human beings or the patients, right? And then applying direct light into the brain may also cause certain heat damage. So these are the ethical concerns why we cannot apply this technique as of now to the humans. But yes, this technique is currently in preclinical research where we are trying to, identify new and new signaling pathways involved in certain disease and how they are getting modulated after the drug injections. Okay. So these are, some of the, pictures, from the surgery which I have done, while performing the surgery. So this is the, macro LED electrode, where you can see the indicator, the light indicator is there. And this is the needle which you can insert, this is looking straight, but this is not straight. You can see it here, right? So this can easily go into the brain in whatever brain region you want to inject it. Okay. There's a flag holder, and then you inject it, you hold it like this with the histotoxic, holder. So instead of Hamilton sling, you are holding the electrode right now. Okay. And then you insert it into the skull of the brain. And once it is inserted, you can again, secure it, suture it, and then, close the wound and then wait for the recovery of the animal. Now this animal is having the electrode inside the brain, and you can decide, which neuron you want to activate or silence. This is like after 1 week, and this is after 4 weeks, and this is after 8 weeks of surgery in these animals. Now this, what you see here, this is me, I visited John Hopkins, in 2017, and this is one of my colleague, Dr. Chen Xiu, basically, who was, working with me that time to, implant this electrode into the spinal cord. That time we implanted this electrode into the spinal cord. So these are the different kind of electrode. It comes in a circular form and in the oval form as well. So what do you see here? It is a condition place preference apparatus, which consists of basically, two different chambers. One is having a stripe, one is having triangles, and their, bottom is also different. One is having a plane surface, another is having a grooved surface, so that animals can identify, okay, these two chambers are different from each other. One thing also you can note down, one of the chamber is wired, while another chamber is not wired, right? So why it is done so? Basically, so that, this chamber can be, controlled by with a certain light in the wavelength, basically, and other one not. Because you wanted to understand in which chamber animal will spend more time, whether after activating the neuron or in the absence of activation of neurons. So this kind of experiment we have performed, that time. So this is the neurolux. This is the device, by which you can control the wavelength of the stimuli or the light. So this was a video, but I think I'm not able to play because I think I don't have the source file. But in this video, I wanted to show you when the animal is, this mice, because this is a corridor and animal can easily go through. There's an underpass here, from which animal can travel from, one chamber to another chamber. So this is the basic instinct. When you place the animal in the corridor, they will move all around the, a chamber basically from one chamber to another chamber. You can see this is different from visual cues. This is also different in terms of, tactile cues. And as soon as the animal is entering this chamber, the light will be activated on the head of the animal. So this is like where the neurons are activated. And if the activation of neurons is giving them the pain relief, because we are working on the pain neurobiology that time, so we wanted to see if we're activating that specific neuron, animals can get the pain relief or not, because these animals were nerve injured, disease animals. So, and then as long as they are moving to this chamber, the light is not blinking, okay, because it is not wired. So that's all from my side today. I would like to, acknowledge my postdoc supervisor, Professor Srinivasa Raja, who is a director, and at John Hopkins University, and also, thankful to all the students in the lab. And Ankit is currently pursuing his postdoctoral research in John Hopkins, Baltimore, Maryland, US right now. He was a senior most student. Akhilesh is currently, the senior PhD student in the lab. So he's about to submit his PhD thesis. And other students are working as PhD and master students in the lab. So thanks to IIT BHU and all the funding agencies to support our research. So he's, Professor Raju, who is my postdoc supervisor as well. He received, John J. Bonica Award in the year 2021. So we had the ISP World Congress in Montreal, Canada in 2022, where he has been awarded this one of the prestigious award. And during that time, he presented this slide to all the, like scientists, which is full of auditorium with thousands of people, and he has, what you can see here, like with the orange color are the people in the, inside of the circle are his, mentors, his teachers, Dr. Rajas. And then the people with the, this purple color are his co-investigators, whom with he worked, over a period of time. He just retired last year. He just retired from his clinical and the preclinical duties. And the green color is the postdoctoral fellows from his lab, which is the outside circle. And you can find, myself. I'm here in the last row. So, yeah. So that's all. I would like to, end this, talk, with just, one small quote: dream big, work hard, stay humble. And thank you so much. If you have any specific question, we can discuss for like another 5-10 minutes. And I think another session is also, handed over to me because one of the speaker, is not able to, come today due to, some personal issue that is there. So we have to start another session from 5:30 p.m. But before we start another session, we can have, some quick questions. And thank you for your patience. Thank you. I'm not sure whether it is directly relevant to your work or not, but I'm hopeful that, this might have been useful for some of you. Okay. Okay, so if we don't have any specific question, today, so yeah, then we can take a quick break of like, 10-15 minutes, and then we can see each other again, at 5:30. Is that okay?
Yes, sir.
Okay, thank you. Then we'll take a break, for like 15 minutes. Thank you. So, good evening, everyone. I hope I'm audible to everyone.
Yeah, Dr. Manoj. Yeah.
Thank you. So I'm sharing my screen with you. Okay. And as I mentioned, this session, is little different, from the previous session. In previous session, we have, discussed, about, histotaxic, which is one of the key technique in neuroscience research these days. In this session, I will, try to cover some of the components which are related to the grant writing or publication writing, why it is important and how do we do that. I think this topic is, relevant to many of us who are in the academia and research, because as an academician, we have to secure the funding, and then we also have to, do the research, and then we also have to communicate our findings for publication in some, good journals. So I think this session, will be of, interest to many of you. Okay. Yeah, feel free to let me know, if you have any questions, please keep it, for the discussion, after the presentation. So some of the, like points which we are going to cover in this session, like why publication writing is important. Many of you might already be having an idea like why we do that, but we will still cover, some of the key points, why it is important. Then we'll also talk about what are the different types of publications, then how to identify the right journal for your manuscript. Next, we will also talk about the plagiarism, which is one of the, malpractice associated with the publication. And, there are a lot of issues which people don't understand, especially in the beginning of their careers, So we will talk about that as well. What are the consequences and how we can take care of that. We will also talk about, like funding, also we'll not talk about funding as such, in this today's session, maybe some other day, but we will only, cover only up to the publication, like why it is important. Okay. So naturalist life is a quote, like a naturalist life would be happy one if he had only to observe and never to write, right? If you just have to observe something and don't have to worry about anything, in science, like it doesn't matter how wonderful and how spectacular your results are. When the work is always, it's not completed until the results are published in some journal. So you have to make sure that you are doing work on the bench, and then you are interpreting your data, and then you have to publish it, for the public domain. So let's explore some of the key steps which are involved, from rough manuscript to the published paper. So planning your manuscript writing. So the first question is why to publish, why it is important, So it is one of the necessary steps embedded in the scientific research process, especially for the beginner. It's very, very important to understand why to publish. So it is also necessary for graduation and career progression. We all know that, for a PhD student, हालांकि अभी recently UGC की तो कुछ guideline आई है जिसमें उन्होंने कहा है कि for PhD it is not mandatory to publish your work, but still, in many of the institutes, especially, like some of the, like IITs or NITs or ISERs or some other institutes, it is still mandatory in India to publish your PhD research work if you want to get your PhD degree. Although it is not mandatory for master's students or like postdoc, but for PhD it is must. If even in IIT BHU, if you have to get graduated and have to get your PhD degree, you have to publish at least 2 research papers, in so Scopus Index journals. Now the next question is what to publish and what is publishable, you can say that. So it is very, very obvious that anything which is new and original in terms of the findings or in terms of new technique or methodology, that is supposed to be published. Any reviews can also be published, or a summary of particular subject. If somebody has done a relevant work in a particular area for over a period of time, that can be compiled in a form of review or mini review and can be published. Manuscripts that advance the knowledge and understanding in a certain scientific field. So of course, those kind of work should be encouraged for publication. Now the next question is what not to publish. Again, it is very, very obvious. The reports or the data which is of no scientific interest generally is not supposed to be published, because nobody bother about that work, or it's of no scientific utility. Basically, it's a crap, basically, you can say, right? So it is not supposed to be published. The work which is out of date, duplication of previously published work, this is very, very important. Most of the time when graduate students join your lab, masters or PhD, we talk about the novelty aspects. We told them, okay, you do the literature review, you figure out whether in this particular area what work has been carried out already, and what are the what are the questions which are still pending to be answered. So that we ask the students when they do the literature review part of their work, just to make sure that we should not duplicate the already done work, because that will be just a wastage of time, money and other resources, and will not add anything in the scientific domain. Incorrect or unacceptable conclusions. Sometimes the conclusions are unacceptable, or you can say they are incorrect. Those also can be avoided. So for all this, you need a good manuscript to present your contribution to the scientific community. So this is why you need to publish, because you have done something which you think is original, novel, you think it can add to the scientific knowledge in a particular area, and people should be aware about it. And definitely it is important for a student to get the degree, and also for faculties, also for the career progression also, it is very, very important. Just like in IIT BHU as well, if you want to get promoted from assistant to associate professor, minimal you have to have at least 3 to 4 research articles in that span of time. So it's required for career progression as well. Now, before you start writing your manuscript, whether it is any kind of manuscript, you need to think about why you want to publish your work. Is it new and interesting? So these are the set of questions which you are supposed to ask yourself. Whether the findings which you want to publish, if it is a research, is it new? Is it interesting? Whether the scientists who are working in that area, will it add to their knowledge? Is it a current hot topic? Have you provided the solution to some of the novel and the difficult problems? So these questions you should ask, and then finally you should ask, are you ready to publish at this point? Whether the data which you want to publish is good enough, or is it completed, or do you think there are other set of experiments or questions which you need to answer before you think that it can be pushed for publication further? If there is yes to all these questions which we have discussed about, then you can start for preparing your manuscript for the publication. Again, if you have to decide, are you ready to publish or not, you can just look after just of these few points. If you think the work is original, or it will lead to significant enhancement of the published work with the up-to-date review of a subject, then I think you think that you are ready to go for the publication. As I mentioned in the previous slide as well, if it is outdated, conclusions are incorrect, if it is a duplication of the previous work, then it's a big no. You should not go ahead and should stop at that point. Now, what makes a strong manuscript? Because so publication is just like a, kind of you want to, sell your work basically. And it has to be done in a manner which should be, easily understandable by the audience. So the manuscript should be very clear. It should be useful, and the message should be very, very clear and exciting. It should be presented and constructed in a very logical manner, which allows the readers to easily grasp the significance. I always tell my students when they start writing the first draft of their manuscript, writing a good manuscript, is not an easy task. You need to understand what are the different components of the manuscript and what are their significance. We will talk about that in coming part of my slides. But I always tell my students, first thing that before you basic 1 second. सर एक लेक्चर चल रहा है, मैं आपको थोड़ी देर में कॉल बैक करूं. So So basically, I always tell my students before they start writing their, first draft of manuscript, I ask them to, to first read the manuscripts which are published in the very good journals, how they are writing. And any manuscript, it's just like a story, or it's like a movie basically, right? It should have a certain, flow. And when you are reading it, from top to bottom, like starting from the abstract, it has to be written in a manner that when the person starts reading it, the person should get motivated to read it more and more. So it has to be presented in a logical manner, in a proper flow, so that the person can, connect one dot after the another. Sometimes people are writing it, in a very jumbled way. Like one paragraph is not connected to another, another is not connected to the next. And then when a person is reading it, they're lost what authors want to convey actually. So it is just like a movie. It's just like a story, you can say, which has to be written in a proper logical flow, so that people can grasp it easily. Any editor, reviewer, and readers all want to receive your message or your work in a well-presented manner. Now, what are the different types of manuscript which can be written? Most of you might already have an idea like what are the different kind of manuscript, but still, for your information, full articles, the original articles, right? So these are the research articles which are also known as original articles, in which you have done some experiments, some bench type work or some computation work, which has involved some experimental work, and you have certain findings which you think can be published. So these are the most important papers because they add to the current knowledge in that particular area. Okay, and often they are substantial and significant, completed piece of research. Okay. Next set of manuscript or next type of manuscript is the letters or the rapid communication, or you can call them short communication. So these are the quick and the early communication of significant and original advances. They are generally much shorter than the full articles. Now, for example, you have done some experiments, okay, and you think that your experiments are, the findings are very novel, which you are supposed to convey quickly to the, people working in your area. So you can write a short manuscript, with some key results in that. So these are known as a rapid communication, which can be published quickly for, for wider domain of scientists, who are working in that particular area. Now, the third type of publication is a review paper. We also call it perspective. Most of the time, good review articles are generally invited by the, editor-in-chiefs or editors of the big journals like Nature, Cell, Science, they mostly invite. So they are, generally, solicited basically. Okay, invited means solicited, and they are generally invited to the people who are expert in that particular domain. For example, if somebody is working in a area of Parkinson, Alzheimer, you can say, any other area for past 10, 15, 20 years. So they will invite that person to write that, article on that particular area. Like, what is your experience or your view in this particular area? You have worked on this area for 10, 20 years. Now, please write, one expert review. So these are known as the expert review. Let me enter you. Somebody want to join. Okay. Now, so these kind of reviews, basically, they are written by the experts in the, area. But apart from that, the reviews are also, written as like uninvited or unsolicited reviews, you can say, right? So these reviews can be done by anybody who is working in that area, and they are mostly uninvited. And there are a lot of journals, medical journals, or which are able to publish those review articles. So even if you're working in a particular area from last 5, 10, 20 years, you can compile an important piece of information from the most recent research done in that particular area in the last 5 to 10 years. So when I was writing my first review article when I was in PhD, I did not had an idea like how a review article is supposed to be written. I was able to write the research articles, but when I started writing the review article, I realized that writing a review is much more difficult than writing a research article, because here you have to compile a lot of information, you have to read a lot of literature in that area, and you need to collect the most updated information. Okay, and then come up with a nice piece of, work which you can present to the audience. Okay. But at that time, I didn't know about the novelty in the review article. When I joined for postdoc in John Hopkins, I joined in 2012 for postdoc, and so I was working on one research project where, we were screening some molecules for neuropathic pain, and these molecules are selectively targeting the microglia. We worked on that project for like at least 6 months to 8 months. And unfortunately, that project didn't work out because those molecules did not show any efficacy, in that neuropathic pain model. But during that process, I read a lot of new articles or previous articles in the field of glia and pain, how glia is involved in the generation of neuropathic pain, all those things. And I was having the most updated knowledge in that area at that time. Then that project didn't work out, so we left that project. But when I went to my postdoc supervisor, to ask him, that I had read a lot of literature on this, area, and I came to know about some of the interesting, or contrasting, like findings which are there, in this area. Some people said that glia is good, some people say glia is bad, but there's some information, which suggests that when they are good, when they are bad. So we can write a very, a comprehensive review on this topic. So yeah, I'm telling you, my postdoc supervisor, Dr. Raja, he grilled me like for at least 45 minutes on that. Why I want to publish that particular review? People don't write review that outside that easily. Only when they think that they are expert in that area, or only when they think that okay, this review can significantly add. So he asked me the very first question: why you think it is novel? And finally, I was able to convince him, how this review is novel, and there is no such review, published in this, area. So this is a new piece of information which we are going to convey to the scientific world. So after 45 minutes of discussion, he was convinced. And finally, we were able to publish that review article in Neuroscience and Biobehavioral Review, which is one of a very good journal in our area. But that is the first time, in my postdoc when I realized that, okay, so novelty is the aspect which is not only involved with the research, but it is also associated with the review article. You cannot just publish the crap, For example, somebody has written on diabetic neuropathy epigenetics, right? Now I'm writing another piece of work, just compiling, the same set of literature or different set of literature. So how my review is different from the already published review, that I have to ask the question before I start writing any review manuscript. Then self-evaluation of the work is very, very important, to understand whether it is sufficient for a full article or your results are really thrilling and important, so that they should be shown as soon as possible to the scientific world. You need to ask your supervisor and you need to discuss with your colleagues as well, for advice. In India, sometimes we are not very open, but I think this should be unfair, people should become more open in terms of, discussing their, findings. People should not feel insecure. Most of the times in India, collaborations are not, very fruitful. And the reason is that people are insecure. They think that, somebody will take their work or somebody will duplicate their work. And that happens sometimes, but people has to be really confident, about their work, about their people. They should have trust on each other. Just like outside, people discuss science in open. Okay. And then you discuss with your colleagues and even your students, you can sit with your student and discuss with them, why you think, this work is really, now in a stage where we can publish it. Sometimes outsiders can see the things more clearly than you. हमारा काम हमेशा में अच्छा ही लगता है, हमें उसमें कोई कमी लगती नहीं है. वही चीज जैसे अपने बच्चे सबको अच्छे लगते हैं, कोई कमी दिखती नहीं है. The same goes for our work as well. So it was always very, very important, to take the opinion and suggestions from the others, including your students, your collaborators, or other people in your department or institute. They will give you some critical suggestions. Based on that, the quality of your work will be significantly improved. Now, it's very, very important, how to find the best journal for your submission. This is the part, where we don't pay much attention because we are mostly focused on doing the experiments, then compiling the data, and then, bringing it in a form of publication. But we don't bother about, which journal we want to publish. There is only one thing in most of our, mind, is the impact factor of the journal. We always run behind the impact factor, My suggestion to all of you, unfortunately, most of the funding agencies, also think like that. And that is something which is pushing everybody, to work like that. But if the work is good, it is more important to publish it, irrespective of which journal you are publishing it. Even if it is publishing in a mediocre journal of a very mediocre impact, doesn't matter. The work will make an impact in the longer run. There are many works which are published, which are rejected by big journal in the past by some very well-known scientists who received the Nobel Prize later on. Their work was rejected by science, nature kind of journals, and they have published that work in a very small journal, and that work received the highest citation over a period of time. So publishing it is more important than bothering about the journal where you want to publish it. But you have to figure out what are the best journals for your article type. For that, you can always look into the references from where you have compiled your review or research article. So those references can give you a list of journals, and then you can make a list priority wise where you want to communicate your article. And then you can also review the recent publication in each candidate journal. You can go to the journal webpage and see what kind of work they are publishing, and then figure out whether you can submit there or not. And you can, before you select the journal, it is very, very important that you ask few questions to yourself about the journal. You cannot publish anywhere. There are several journals. I would tell you that every day I'm receiving at least 3 to 4 emails from different journals all over the world, and they're asking you to submit your work in their journal, publish in their journal. Some are open access, some are free, but because they are new journals maybe, and some are even predatory journals. So you have to be very careful where you're publishing your work. You should keep away from the predatory journal. We'll talk about that, what are those journals. Okay, so first question which you have to ask is whether the journal is peer reviewed or not. Second, you have to figure out who is the audience of the journal. Sometimes the findings are really good, but if you're publishing it in the irrelevant journal, it will not hit the right audience. There are certain journals which are highly clinical in nature, like JCI, Journal of Clinical Investigation, which is the big journal in the field of clinical sciences. But mostly clinical researchers read those journals. But research findings do also get published in that journal. Sometimes clinicians are interested in the recent findings, what is happening in terms of clinical research there. You also need to check what is the average time to print. If the journal is taking like 6 months, 8 months, or 1 year, you don't want that kind of journal, right? You want something which can publish in if not too quick, but at least in a period of like 2 to 3 months. It should be online, your article. And then, journal impact factor is important. I always say that don't run behind the impact factor, but yes, impact factor is one way to judge the credibility of the journal. And how the impact factor is calculated. It's calculated based on the citation of the papers from that journal. So if the papers in that journal are highly cited, then the impact factor of that journal will be definitely more. So that defines somewhere about the quality of the journal. But again, this parameter is manipulated a little bit. So we will not go into that aspect right now. But yes, this is one parameter, but it's not very important. More important is to publish somewhere in good journal. Okay, then decide on one journal. Do not submit to multiple journals. This is a very, very important point, which is related to the ethical concerns related to the publication. Nowadays, everybody is in a rat race to get more and more publication, to get more and more citations in a short period of time. But ethically, that is wrong. If you go to, if you just look at the scientists who have really done good work, in some of the, topmost places in, in India as well, and other places as well, in US, Europe, or other places, they have never been in the rat race of, increasing their number of publication. They were never in a rat race of increasing their citation. If the work is good, the citation will automatically grow over a period of time. So focusing on quality is more important rather than quantity of what you are publishing. You're just publishing a crap and then increasing number of publication in your CV, that can help you temporarily for a period of time. But in the longer run, that is never good for your career and CV. Because ultimately you will realize that you have published crap and you have not contributed anything to the science. So it's very important. So decide only one journal. Communicating in more than one journal is at a time is unethical. Not only unethical, it is an academic crime. Okay, so do not do that, and always advise your students also not to do that. So now this is, one tool which can help you to choose the right journal for your work. So there are several ways you can, basically type the keywords related to your work, and we'll give you a list of journals, where you can basically communicate your work. Also, you can basically copy paste your abstract based on your, abstract. This tool, Elsewhere tool, will start matching with the journals related to that abstract, and will give you a list of journals where you can communicate your work. Also, you can also search for a journal by name if you already have an idea. So there are a lot of, like you can see, we accept and publish more than 250,000 journal articles. This is the quanta of work which is being published by Elsewhere alone. 250,000 every year. And there are a lot of other publishers just like Elsewhere, Taylor, Francis, Willey, and lot more, right? You can just imagine, the information is being published at such a, high rate. Now, the question is, what makes a good manuscript? Why you should, worry about that your manuscript should be good? Sometimes we are just trying to, publish as quickly as possible, and we pay less attention to what we have actually, written. So be very, very careful what you are writing, and make very sure what you are communicating to the journals. Okay, so your manuscript should contain a clear, useful, and the exciting scientific message, which is presented in a very, good flow in a logical manner that the reader can follow once they read your manuscript. You also need to make sure that it is formatted to the best showcase the material. So formatting plays a very, very important role, and all the different journals, they have their own formatting guidelines in Guide to Authors on their web page. Then it should be written in a style that transmits the message very clearly. As I told you, it's like a movie, it's like a story, how you're presenting it. If the director is not good, you're a director of your story, right? So you're a director of your manuscript here. You're a corresponding author or a first author. So you have to write it in a manner so that it can easily be, understood by even the layman. So you have to keep a balance between the layman language versus the technical language you're using in your manuscript. Now, structuring your article, what should be the different components, how you should structure it. These are some of the components,
Dr.
Yes.
Tiwari, just,
Yes.
sorry to interrupt. My apologies. I just sent some message in the chat, if you can tell others, because, for the career advancements, it's very crucial, So if you can tell them, of course, I know that, but if you can tell them others. Yeah. Thank you.
Thank you so much. Thank you so much, sir.
Yep.
So, So, Professor Arun has mentioned about, like Q1, Q2 journal. There are different category of journals, and the UGC has also given a list of journals, like, where you can publish, and for a career advancement, it is very, very important. So there are different, journals which are considered in the, I think the top, like 25%. So top 25% journals, comes under the Q1 category basically. And then the journals which comes after that, they comes under the Q2 category. So journals have been categorized, and you can go to the journal web page, to understand, to which quartile they belong, Q1 or Q2. So it again depends upon the, again impact factor of the journal or other things.
Yeah, because, after 2018, June probably, 18 or 19 June, it's very important that the journals are listed in the UGC care, and the Q1, QT, and Web of Science, and so on. Yeah.
Right, sir. Right.
So let them be very well aware of that fact.
Thank you, sir, for bringing that point. Yeah.
Thank you so much.
Yeah, just to add up to that, to your comment, sir. So people should be looking up at Q1, Q2, Q3, or Q4, journals, and then UGC Web Care, so which has 2 groups. Initially, it had 4 groups.
Yeah.
Right? Now it has been categorized into 2 groups, right? UGC Care 1 and 2, so on and so forth. Group 1
Absolutely. And UGC care is more important.
more important these days, right? So you should be looking up at that. That's fine, and we can discuss that in the discussion section.
Thank you for bringing that point.
Yeah. And then quarterly journals, what are bibliometrics and all that, we can discuss that in the discussion section.
Wonderful. Yeah. Thank you,
sir.
So so if we talk about the structure of the manuscript, we already have some of the experts with us. I'm really glad to see that. But yeah, for the beginners, or those who are, still learning, how to, do it. So we have certain components in any of the manuscript, ranging from title, abstract, or the keywords, and then, we have the introduction part, methodology, result, discussion, and then, the last part is about telling your story based on what are your findings. So that can come under the conclusion section. And then after that, acknowledgement, references, and then supporting information or the supplementary data you can talk about. Now, if we talk about title, how to, decide the title of a manuscript, it is very, very important, like when you are debating on the title of your manuscript, many times people take it very casually, but title is something, which will make a big impact on the reviewer, where journal is going to be reviewed. And once it is published, so that will tell about the kind of work which you have done. So it should identify the main issue of the paper or the problem which you have addressed. Second thing, it should be very, very concise. It should not be too long. It should not be too small. It should be accurate, unambiguous, specific, and also complete. Most importantly, it should use the professional language, and we should avoid using the abbreviation in the title. This is another mistake which people do. They use some abbreviations, which are approved, which are authorized, but you cannot use any abbreviation in the title of your manuscript. Most importantly, it should be able to attract the readers. It has to be short, and with the title which are short and catchy, they sometimes, most of the time, they often gets better cited. The message should be clear. So there are certain examples, like this is one of the title of, like which one has given to one manuscript, action of antibiotic on bacteria. So the title looks very vague. The message is not clear what author want to communicate, right? So we can refine it in a more scientific manner, like inhibition of growth of Mycobacterium tuberculosis by streptomycin. Now the message is more clear. So we are talking about a specific antibiotic, which is streptomycin, and then we are talking about, a specific disease, like which bacteria, Mycobacterium tuberculosis. So it has to be more elaborative in a way so that audience can understand what kind of work you have carried out. It should not be redundant. Then keywords are the another important, prominent feature of your article. Most of the time authors again don't pay much attention to it. They just copy one of the few things, which are in the manuscript, and then quickly put it. But again, these are the labels of the manuscript, and they are used by indexing and abstracting services. It has to be very, very specific. For example, I work in the field of chronic pain neurobiology. So if I worked on the chemotherapy-induced neuropathy, so my keyword should involve, neuropathy or pain or chemotherapy, these kind of keywords. If I use some specific drugs, so that drug can come as a keyword in the keyword. And it should use only established abbreviation. That is another very important point. You can use abbreviations here, not in the title, but it has to be established, not any like which you are giving by yourself. Okay. And check the guideline for the authors before you, proceed for it. Now, abstract, another important, a part of your manuscript. Try to keep it as brief as possible. Most of the journals, they have their own guidelines. Some journals, they ask for the structured abstract, where there is certain pattern which they want to follow you. For example, some journals want you to, break your abstract into 4 components, like, rational, like objectives, then methodology you have used, and then discussion conclusion. So in 4 or 5 different components, you have to break your abstract. That is known as the structured abstract. But there are certain journals which ask for the unstructured abstract. Unstructured abstract means they want you to present or summarize your findings or your work in just, 1 paragraph of like 250 words or 300 words. So it depends upon the journal where you are communicating your work. So, but whatever it is, whether it is a structured abstract or unstructured abstract, the main motto is to summarize the problem, the methodology you have adopted, and the results which you have, obtained while, carrying out those experiments, and overall conclusion from that. And make sure that it is clearly written and easy to understand. Sometimes you don't have to write like all the results here. That is not allowed. Otherwise, it will make your abstract redundant. And make sure that it is accurate and specific. You don't have to, claim anything like which is beyond your findings. And while also it has to be catchy, so that all readers can feel motivated to go into the full text of your article. Most of the time people go to the PubMed and they put the keywords, and when they find that these are the 50 articles or 100 articles in that area, they will only try to go to the full text of that article, in which they find the title and the abstract more catchy, okay, and which is more relevant to their work, of course, and write last so accurately so that it reflects the content of the paper. So this is a general rule of 10. If you follow that rule of 10, your abstract will be more, I would say, catchy in terms of presentation. So 1 to 2 sentences should be about the aims and objective of your research. The next 2 to 3 sentences will be about the materials and methodology, and after that 2 to 3 sentences will be about the result part, and last 2 sentences of the abstract should talk about the discussion and the conclusion. So if you follow this rule of 10, your abstract will be kind of balanced, which can give a clear picture about what you have done and what you have identified. Now this is the next part of your manuscript, another important part of your manuscript, which is introduction part basically. So most of the time, people just, don't understand, that why this introduction part is important. And sometimes they put the content which is relevant for the discussion part or conclusion, like discussion part, and they put it into the introduction. But one has to understand, in the introduction, you have to highlight your problem or the area on which you are working basically. What is the gravity of the situation, and what is the area, a problem which you want to solve. And then you also need to discuss about the limitations in that particular area. That should also be the part of the introduction. If you're working on certain disease, you should talk about why this disease is so significant. And you should also talk about the current, treatment approaches which are available in the clinic. And then you should also talk about the limitations of those, treatment approaches which are available in the clinic. And then you should talk about how you're going to sort out that problem. Why your, approach will be different, and from others, and how to solve the problem. And some part of hypothesis can be the part of introduction, and then you can also mention about this is we have already covered existing solutions and limitations. Then next comes the methodology part. Most of the time people start copy pasting the methodology from other publication, but you need to understand plagiarism plays a very, very important role here as well. You need to make sure that the content, of course, the methodology cannot change. If you have use some assays which are used by some other lab, the same assay you are going to use, you have used probably in your experiments, but again you have to reframe the language which you are using. You cannot directly copy paste from others. But methodology is mostly about how the problem was studied, what techniques you have used. You will also talk about the you should not describe previously published procedures, but you have to cite clearly what methodology they have used, and you have adopted it, right? And then identify the equipment and the materials which are used. Use proper notations including chemical formula and symbols, and don't forget to present the controls which you have used in your study. Then next comes the results part. So result part includes only data of primary importance. So what you see in this slide is a huge iceberg, and the tip is above the ocean, and more than 80 to 90% of the iceberg is beneath the sea, beneath the ocean, right? So what does this indicate? The upper part of this iceberg is the data which is of primary importance, and beneath all this data there is a lot of supplementary data which is there, which is a data of secondary importance. So all the secondary data can come in the supplementary file, because journals, they have their page limitation. They cannot publish beyond certain page numbers. So they will ask you to put only maybe some journals may ask you to put only 4 figures or 6 figures. Some may allow 6 to 8 figures, but not more than that. So the other figures which are of secondary importance, they can come into the supplementary file. And then you can also use the subheadings, to keep the results of the same type together, and also try to avoid the redundancy in your data presentation. It is very, very important to use the figures and tables for the efficiency and the clarity. You can write your data, in the form of paragraphs and sentences, but who is going to read those long paragraphs? So there is an art, how you should present your data. So figures are always a good way to present your data in a pictorial manner, which is easy to understand. If the audience will look into it, they don't have to read everything in the paragraph. They can just understand in few minutes what is happening. Whether under certain disease condition, some markers are upregulated, and after drug treatment, whether they are downregulated or not. Some genes are overexpressed, some genes are underexpressed. So those kind of information can be quickly, understood by the figures. Similarly, there are certain kind of data which can be presented effectively in the form of tables. And the most important part is the statistical analysis. It has to be mentioned very clearly in the methodology as well, and in the figure legend as well. So every figure has a figure legend, and every table also has a legend or a title. But statistical analysis is very, very important. Until and unless the data from the two groups are not significantly different from each other, the data you cannot say is different from each other. Most of the time, new students who are learning the research, they say, sir, there is a increase. But once they apply the statistical analysis, it is not significant. So until it is not significantly different from each other, you cannot say whether something has increased or decreased. So for that, there are different statistical tools which are available. Various softwares are available. Then this is how you can make your results more appealing. The caption and the legend should be self-explanatory to enable the figure to stand the alone. So it's very important what titles, what captions, what legends you're using. You can use maximize the visual versus the space, and then use color only when it is necessary, otherwise not. The graph should not be crowded. It should be uncrowded plot. You can restrict the data sets. Use certain symbols if you want to distinguish between the different groups, like in terms of significance, whether they are different from each other. Use small symbols like star mark or hash mark, and then use a p-value to denote whether it is p less than, p means probability, less than 0.05 or 0.01, or p less than 0.001. And then, access levels has to be very clear. Label size should be properly, indicated. Then in case of photos, scale marker is very, very important. For example, in this figure, scale marker is mentioned as 500 nanometer. In certain histopathological, images or immunohistochemistry images, we don't know what is the scale. That data is of no use if you don't put the scale marker. Most important thing, do not manipulate the image to enhance the result. This is the biggest mistake which the students or the beginners do while presenting their, result. This comes under manipulation. Even in the Western blotting data, where we, measure the protein expression of certain biological markers in certain diseases, So we perform the Western blotting and we have the data in terms of band. And sometimes students think that background is coming and they try to remove the background. But with the background, they are also changing the intensity of the bands of the proteins. And it is not permissible as per guidelines. You keep it as such, and then you publish it as such. Even if the bands are dirty, images are dirty, they will be appreciated by the reviewers and the scientific community because they are original as such. They are coming. Sometimes people can unintentionally harm their data, and sometimes people do it intentionally as well. They do manipulation, which is not acceptable at all, by using Photoshop and all other features. So those kind of things should be strictly avoided. So for example, this is a data set which somebody has written in a form of long paragraph. So do you think this kind of data set, somebody has time to read? No. So if you can put it in the form of table, it will be easy to understand and quickly one can understand it. So if this kind of data is there, it does not, need a table. This you can write simply in the form of one paragraph. But if this kind of, complex data set is there, then it definitely needs a table. So you need to figure out what kind of data is there and whether it needs a table or not. These are just two lines, this can be presented in a form of one paragraph only. Then discussion is another important, component of your manuscript, which involves, like what results you have got, and then how you're interpreting your data. Sometimes interpretation makes a big difference, The same thing, one see from one angle, it looks different, and other person sees from other angle, it looks different. So in the light of existing literature, what your data is saying, you have to interpret it very, smartly and very accurately also. And you have to make the discussion, part corresponding to the results and complement them with reference to the current literature. So you have to compare the published data, which is published by others in that particular field, and then compare it with your findings, like how it is aligning with the existing literature and how it is contradicting with the existing literature. That also you have to discuss. Most of the time, people just think about the positive, and positive, that everything positive, but that is not something, which happens in science. There's many times when you get the negative result. There's many times when your findings are, are opposite to what others are, are suggesting. So have that courage, have that boldness that you can go against the existing literature as well in terms of certain, parts of your, research. And then, take a stand on it. Sometimes it can give a direction, completely new direction to that field. Avoid the statements that go beyond what the results can support. So don't, overclaim your things. So most of the time we generally use a term may. The drug X may treat the disease Y. So we don't say that drug X treat the disease Y, because that is like overclaiming. So you have performed certain set of experiments, you have certain results with you, and they are just, suggesting that it may happen. So be modest while, discussing your data. Avoid non-specific expression, be highly specific in discussion part. The new terms which have not already discussed in your paper, avoid using those terms. Speculations on possible interpretation that are not rooted in the fact, that also you should avoid. Now in the conclusion part, it's very important, like how you are, putting it in front of the audience, how your work has advances the present state of knowledge in that particular field domain that you are supposed to, discuss in the conclusion part. You should not repeat the results in conclusion, because here you are just making a conclusion based on, what you have found and what you have already discussed in the discussion section. You should not repeat the abstract part even. And here you can discuss, like uses of that particular technology or particular drugs which you have identified and discovered, or you can also discuss the extension of that particular discovery, how it can be extended or applied, or other applications of that particular discovery, how it can be utilized in the future. And you can also suggest certain future experiments in the conclusion part. And be very, very clear, so that it can help the reviewer and editors to judge your work and its impact. Somebody want to join us? Okay. Okay. Now, so it's very important, the conclusion should be very clear so that reviewers and editors have an idea, like, what your work is and how it will impact the, current state of knowledge. And then acknowledgement, this is another important, part of your manuscript, which people, ignore, most of the time. They don't take it very seriously. Ritika, I want to ask some questions. Ritika, can we keep the questions for the later part of the, like discussion? Because, I think we still have some time left with us. And you please keep your questions for the end of the presentation and we'll discuss it, okay? So in acknowledgement, it's very important to acknowledge, like the people who has helped you in carrying out that research. And so you have to include the individuals who have assisted you in your study, like your, advisor. For example, if somebody has advised you on certain set of experiments, you can put their name in the acknowledgement section. Definitely funders, funding agencies, or the financial supporter, that also you can indicate in the acknowledgement section. Proofreaders, if there are certain, for example, in certain, institutes, they have, like grant writers or publication writers, so they should not be the authors. People generally think that, they can be included as the author. No, they have proofread your manuscript. They cannot be the author in your work. They can come in the acknowledgement section. So that's the most, I would say, pity part. We don't understand whom to include as an author and whom to include as a in the acknowledgement section as a contributor. Until unless somebody has not contributed in the experiments, you cannot have them as the author in your work. But that is another kind of unethical practice, which people have, are following basically, just to increase the number of publications. That should be avoided. Okay. And then typist also, if he has helped you in typing your work, then suppliers who may have given the materials or the gift sample from some lab or some institutes abroad, all these people can come in the acknowledgement section, but they should not be the authors. References, this is the part where people do most of the mistakes because they are not careful. So they are found, more mistakes are generally found in the reference section than any other part of the manuscript. It is one of the most annoying problems and causes great headaches among the editors. So these days, it's not a big issue because we have a lot of good softwares in the form of reference manager, reference works, and note, and so many, which can help you to, cite the work in a, more appropriate manner according to the journal style. But make sure that you cite the main scientific publication on which your work is based. Do not inflate the manuscript with too many references. It is very, very important. Good journals, they always avoid too many references. They will ask you to stick with only 40 to 50 references if it is a research article. Good journals, good research journals. If a journal, they will never appreciate more than 50 references. If it's a review article, again depends. But it's a major review or a minor review, again the reference should not go beyond like 150 or 200. Okay. Another important point, which people, don't care or they intentionally do, they put a lot of self-citations. As I told you, people are running in a rat race, unnecessary rat race. But a day will come, I'm telling you, if not today, maybe in coming years, when people will figure out a way to figure out a distinction between the self-citations versus the real citation. Then the people will be in trouble who are running behind the self-citation. So avoid this practice, Don't run in the rat race, don't run in the race where you are trying to increase the citation. Because if the work is good, it will be noticed and it will be, cited by the people all over the world. But yes, there are certain, work which you have done in the past, and if the current work is the, is based on that past work, that you should definitely cite. But never cite too much because it will be, easily visible how much you are, putting in terms of self-citation. So also avoid excessive citation of publication from the same region as well. Have a geographical, uniformity as well. Okay. Now next comes, submitting your paper. Like once you are done with, writing your manuscript, now it is ready. The next big challenge, especially for the beginners, where to submit it, right? So you can see this student, writing first draft, second draft, and then revising again and again. And then finally, when she's tired, then the supervisor says, okay, now it is, ready for the publication. So the cover letter is a very, important part again, which speaks, about your work. Although nobody is going to reject your work on the basis of poor cover letter, but it is just like an application. You are selling your work, in the market. So it's like a job application letter, you can say, right? So most importantly, you should not summarize your manuscript or repeat the abstract in the cover letter. You should only mention what would make a manuscript special to the journal. You can mention, why it is more relevant to get published in your journal, so that editor can take a call whether this work is suitable for their journal or not. Okay. Most of the editor will not reject, as I mentioned, because of the poor cover letter. But yes, it may definitely accelerate the editorial process for your paper if it is written in a good way. Conflict of interest is another important point for, regarding which most of the people are not aware about. It can be a direct financial, conflict of interest. For example, if we talk about certain, like if it is because of job or stock ownership or grants or related to patent. So those kind of, things should be declared in this column. Okay. Then there are indirect financial conflict of interest if you have received some honorarium from some agencies, from some companies. If you have received certain consultancy, it is very, very important to disclose it in the conflict of interest section, because sometimes, you don't know whether somebody want to get a certain set of study to get it published for the benefit, because they want to promote that product. So if it is unbiased, it has to be disclosed in the conflict of interest. Okay. Then sometimes it can be related to career or, promotion or some rivalry, or institutional or sometimes personal beliefs. So there are a lot of other issues, which can, affect the things. So you have to indicate it in the conflict of interest statement. Now, next comes the peer review and responding to reviewer. You can just see here, this is you here with your manuscript ready for submission. And what you see here, like a lot of, people, ready to, handle you with a lot of, weapons in their hands. So they are reviewers and editors, team of reviewers and editors, which are there to make sure that you should not, reach to the end. If you will reach, that means your work is really good when it will be accepted for the publication. Okay. And the main motto is to make sure that the work is improved, and it should be, in a proper format, which can be, assessed by the audience. Then what actually happens when you start submitting your work as an author, if it is meeting the basic requirement, editor will say yes, and it will send this manuscript to the reviewers, assign it, right? If no, the editor will reject it, or it will ask you to revise it. You will revise it again, you will submit it again, right? If the work is assigned to the reviewers, the reviewer will review it critically, and then they will give the recommendation whether this work can be, accepted or rejected. So based on the reviewer's recommendation, editor can take a decision whether to send it for major revision, minor revision, to accept this work or to reject it work. So this is the overall process, once you have done with submitting your work. Now, what reviewers actually look when you submit your work for publication? Reviewers mostly look for the hypothesis, whether the hypothesis is significant, whether it is novel, whether you are answering a important question or not. They will also look for the originality of your work. Then they will also look whether it is making a clear progression, in that particular domain, and whether your findings are well presented or not. So you can see this graph is a plot between, technical quality versus novelty. If there is certain works which are not good technically and they are not novel as well, they'll be straightforward rejected by the editors. If there are certain work which are highly novel, but they are not having a good technical quality, they are question marked, may or may get accepted. In the similar manner, if there are certain work which are, having a high technical quality, but poor novelty, they are also under question mark. They may be accepted or may not be. But the work which is high in terms of novelty and high in terms of technical quality, the chances of their acceptance is always pretty high. Now, once you get the comments from the reviewers, how you should address them, this is an art. Again, most of the people, they don't know how to address the reviewer comment. Mostly people, they don't give the rebuttal. They mostly think that, if they give the rebuttal, reviewers will reject their manuscript. But this is not the case. Sometimes you have to fight with the reviewer as well. If you think what you have done and what reviewer is pointing out is not correct, you have a right to rebut it. You have to right to take a stand against it. And you have to convince the reviewer and the editor-in-chief that you are right. But sometimes, how you're responding, it's very, very important, It's an art, how you're responding. So, for example, a reviewer is, sending a comment that, okay, the author failed to reference the work of Smith et al., who solved the problem 20 years ago. You're not supposed to respond like, we didn't think anybody had read that. Actually, their solution is better than ours. So this is a non-scientific way of responding to reviewer. You cannot also write, yes, we know, we thought we could still get a paper out of it. So this is redundant way of, responding to reviewer. I know nobody will do that, but this you should understand, you have to write in a more scientific and technical manner. So you can write in a way like reviewer raises an interesting concern. However, our work is based on completely different first principles and has a much more attractive graphical user interface. So you can address the same question in a much better manner, which can, address the reviewer's concern as well. So again, rebuttal part, as I told you, if you think that reviewer concern is not, correct, and then also you can take a rebuttal and then you can write to them that this is what we feel, what we believe, but in a proper logical manner with some proofs and evidences, not like in an arrogant manner. These are some of the reviewer assignments, I'm sharing just with you that we received like this, that they ask us to review the different, manuscripts, and then we have to respond to them as a reviewer. And sometimes as an editor, we have to assign it to different reviewers in different journals. Sometimes you are receiving too many work, to review, so you don't have time. So before you accept to review any work, make sure that it belongs to your domain. Most of the time, editors will contact you based on your expertise, but sometimes you may receive the irrelevant work as well to review or to add it. So you can take a call whether you want to review or not, and based on availability of your time. These are some of the publications, from our lab, which has been published in some of the good journals like Nature Neuroscience, then Pain, Anesthesiology, ACS Chemical Neuroscience. These are some of the, key works from our, in chronic pain neurobiology and development of novel therapies for the treatment of different pain disorders. Then this work was published in, PNAS, Proceedings of National Academy of Science. And this is another work which is on chemotherapy-induced neuropathic pain, which is published in Life Science in 2022 only. So with this, I would like to thank, all the students in my lab, who are the real contributors, for any research we are publishing, and thanks to the different funding agencies as well, including, support, infrastructure support from the IIT BHU Varanasi. And I thank all of you for your time patience, in listening me for like almost 3 hours. So thank you so much. If you have any specific question, I'm happy to address.
Yeah, great talk, Dr. Tiwari. Great talk indeed.
Thank you, sir. Thank you.
I have 3 small points to add.
Yes, sir.
I'm sorry if I would have missed it because I was back and forth to the head office and also.
Yes,
so, for those who are in the scientific area, like in the science areas, whatever, IT, medical, or agriculture, whatever it is. So you got to be mentioning the names and the the company of the good instruments you have used for your scientific experiments. That also helps a lot. If you have used the maybe internationally known experiments, instruments, that will help you. Second thing would be like, the software which you are trying to use for your analysis and so on. That would be very helpful. And third thing, you already mentioned there, the language, if we can get it edited by somebody, some colleague in the Department of English or whoever has good English or whatever, I'm not like promoting English or like, commenting on Hindi, but the thing is like, if you want it to be published in the good journals, you got to be having good language there. Please see just a small add to your talk. Excellent talk again. Thank you so much.
Thank you, sir. Thank you so much for these important, I think points. I think it's very helpful to the audience.
Thank you.
Any other questions, And please fill the feedback form. I would really appreciate if all of you can find, some time, maybe just few minutes, to please fill the feedback form. That will really help me to understand, how we can improve in subsequent sessions, or, like how do you perceive this, today's two sessions. So if there are no further questions, then, I think, feel free to write me, if you have any. There's some, please share again the link I got. Okay.
Yeah, I would like to meet you sometime.
Thank you, sir. It will be my pleasure, sir. Thank you, sir. Thank you.
Thank you so much.
You,
excellent talk. Yeah.
Thank you, sir. Thank you. So, okay, then, feel free to write to me, or if you are visiting Varanasi sometime, feel free to give me a call or, visit the office. Well, we're happy to meet you. Thank you so much, everyone.